I'm Sho Watanabe, postdoc in Department of Bioengineering at Rice University. Today, I will talk about our new technology that enables non-invasive monitoring of in vivo transcriptions in living animals. Our laboratory is mainly working on the development of non-invasive neuroengineering tools. The motivation behind this is to develop translatable tools to safely and ethically study intact brains in large animals, which should definitely lead us to identify new therapeutic targets. However, at present, we don't have a good tool, especially for molecular studies. For example, transcriptional analysis like qPCR or RNA sequence they are already critical in biomedical research. However, all of these technology can apply only to postmortem tissues, which prevent us from monitoring the gene expression dynamics over time in the living animals. This is a problem because personally, I want to understand how my disease progress while I'm alive. In addition, now there is no good scalable, highly multiplex systems in vivo like RNA sequence. So the purpose in this study is to develop non-invasive tools to achieve transcriptional analysis in intact brain, ideally that has a highly multiplex systems. So we focus on the serum markers. Why serum markers? Serum marker is non-invasive tool for monitor tissue physiology. However, naturally secreted serum marker give us only few biological information. So our idea is, why not making a synthetic serum markers that tell us any information we want? Based on this concept, we developed the synthetic serum markers called RMA. RMA is a reporter protein, but has the capability of crossing blood-brain barrier. Using these RMA reporters, we created new platform that allow us to monitor the mRNA expression with a simple blood test. Here's a molecular principle. We leveraged the recently developed RNA sensor technology to directly sense the target mRNA to produce RMA expression. Once RMA produced, it immediately secretes extracellular regions and cross blood-brain barrier and release into the blood. So serum RMA level should be correlated with the intracellular mRNA level. So proof of concept, we demonstrate the intact can detect the exogenous gene, in this case TD tomato, over 2 weeks and 3 weeks. And we show intact has a very high specificity. And next, we try to monitor the endogenous gene expressions, in this case ARC, which is one of the neuronal activity responsive genes. So we transduced ARC sensors in the hippocampus and chemically induced the neuronal activations, and we observed the significant increase of RMS signals in response to the neuronal activations. So here's more exciting data. We try to monitor the gene expression at 3 brain regions. We prepared 3 RMA reporters, which can be orthogonally detectable by using different luciferase. And we observed the serum RMA profiles that is corresponding to the gene expression pattern at individual brain regions. So in conclusion, Intact is the first technology for non-invasive monitoring of mRNA expressions that allow us to detect both exogenous and endogenous transcript. As a first step for highly multiplex system, we we demonstrate Intact can monitor the gene expression at 3 different brain regions. As a next step, combining with the protein barcoding system, we expect that we can make the highly multiplex systems, which may be called non-invasive transcriptomics. And we keep working on the sensors, which should be helpful in neuroscience. Thank you for your attention.